Welcome, and thank you for attending this webinar, where we introduce the MxP Quant 1000 assay, the latest metabolomic solution from biocrates. MxP Quant 1000 sets a new standard of excellence in quantitative metabolomics profiling that is on the verge of transforming medicine. This new assay was developed with one of the most pressing global health challenges in mind: chronic disease. So I'll begin with a few words about chronic disease and the unique perspective provided by metabolomics to solve this enormous burden. I'll also introduce the concept of 5P medicine, a framework designed to help harness the innovative power of molecular tools such as metabolomics for modern medicine. I'll conclude with 3 case studies showcasing how to apply metabolomics to screen samples, leverage data, and translate insights in multiple disease contexts, using, of course, metabolites measured with the MxP Quant 1000 assay. Next, my colleague Markus Langsdorff will tell you everything there is to know about this exciting new assay and how you can work with biocrates to apply metabolomics in your research. We've reserved 15 to 20 minutes at the end of the webinar to answer all your questions. So let's get started with this ambitious but essential goal to solve chronic disease with metabolomics. In 2013, the WHO issued a global action plan for the prevention and control of non-communicable disease by 2020. As you can see on the slide, whether you look at diabetes, dementia, or cancers, these actions did not manage to curb the tide. And by 2021, globally, 75% of non-pandemic related deaths were attributed to chronic or non-communicable disease. In 2023, Biocrates published a white paper diving into the origins of chronic disease, from classically metabolic diseases such as metabolic syndrome and type 2 diabetes, to conditions with strong environmental determinants like cancers, autoimmune disease, and neurodegenerative disease. In this document, we outlined the critical role of diet and of its effects on our gut microbiome in the etiology of these diseases. What you can see represented on the slide here is that the imbalance in the gut microbiome and the different nutrients and metabolites that come from the diet have an impact on health through the action of these metabolites that you see represented here, that are going to drive inflammation, that are going to cause mitochondrial disturbances and organ dysfunction, and progressively lead to an imbalance in the body and to prime the body for chronic disease. So the strong influence of our environment and lifestyle makes it crucial to study chronic disease with the omic technologies that can sense the impact of our environment on our health. The scientific community has invested great effort in the last 30 years in applying genomics to study chronic disease. And from this work, we've learned that only about 10% of the variants related to any of these diseases can be attributed to genetics, roughly. The rest, that's governed by the way we live our lives. So for me, this means that the remaining 90% of variants are begging to be investigated with metabolomics, not only for diseases like cardiometabolic conditions, but also in oncology, in neurology, and other specialties. In fact, combining genomics, metabolomics, and other omics is a great way to unlock extra information by bringing the pieces of the biological puzzle back together in multi-omic studies. If you're interested to learn more about how to integrate metabolomics with other omics, we'll get back to this towards the end of the presentation. But for now, let's get back to metabolomics. The team of scientists behind our white paper also built metabolic models that you see represented on the screen here for each one of these diseases. And these models focus on the metabolic characteristics of the disease and the mechanisms behind their changes. So for each condition, a clear link to microbiome and microbiome metabolites could be established, drawing attention to chemical classes such as fatty acids, particularly short-chain fatty acids, bile acids, and indole derivatives that can all come from the metabolism of the gut microbiome, but also energy metabolism intermediates, urea cycle and nitrogen metabolism metabolites involved in immune regulation, and also many, many classes of lipids with both signaling function and structural properties. These insights were at the heart of our selection of metabolites for the MxP Quant 1000 panel, which is expanded with a broad set of core metabolism metabolites. But in order for metabolomics to have an impact on medicine, the results must be easily translated into actionable insights. So at Biocrates, we've put much effort into democratizing the interpretation of metabolomics results. Next, I want to introduce a framework that encompasses the myriad ways that metabolomics can be leveraged to transform medicine. 5P medicine is a concept designed to combine the steps in the application of molecular tools, such as metabolomics, to build the medicine of tomorrow. So whether it is for preventive, predictive, or precision medicine, for population-based medicine, or for participatory medicine, metabolomics is a powerful tool to develop solutions that take into account our environment in the study of health. I want to walk you through a few examples of how metabolomics is used in 5P medicine. You see many of them listed here on the slide. For example, given the role of diet and exposures in chronic disease, metabolomics is a perfect tool to devise strategies for the prevention of these diseases. For example, with dietary or other interventions that come in early, even before or in the very early stage of disease, and also monitoring tools based on metabolite levels that can help to monitor the response of an individual to the intervention. Biomarker discovery is probably the best known use of metabolomics in predictive medicine, while metabotyping or stratification of individuals based on their metabolic profile is a recognized tool for precision medicine. At the population level, metabolomics is the ultimate tool to study environmental variance, although it still remains the missing piece in many multiomics puzzles. And last but not least, advances in at-home sampling combined with quantitative and reproducible metabolomics that make the comparison to reference ranges possible are enabling patient-centric solutions through participatory medicine. In the next part of the webinar, you will see numerous ways in which metabolomics has been applied within the framework of 5P medicine. I hope that these case studies will inspire you to use a similar strategy in your own research. So let's get started. It's now time to discover the 3 case studies that were handcrafted by our team to showcase how metabolomics is used to screen samples, leverage data, and translate insights into the innovative solutions that are needed to advance medicine in the context of the 5Ps. As we dig through these examples, you'll also get a first glimpse at some of the new metabolites, chemical classes, and pathways that are covered in the MxP Quant 1000 assay. And before we dive deeper into the metabolites and pathways, I want to take one more minute to highlight the structure that is going to run through our case studies today. Applying metabolomics with the 5P framework can be summarized in 3 simple steps that I've already mentioned a couple of times, so you might know where I'm going with this. Step 1 is screening samples. So you analyze your samples with broad metabolomics and lipidomics profiling. With Biocrates solutions, the result is a quantitative and quality control data set that is perfectly suited for step 2, where you leverage data. This can be done in several ways to get closer to the biological meaning of the chemical changes you measure with metabolomics. And when you work with the MxP Quant 1000 assay, you can gain instant insights, for example, thanks to metabolite sums and ratios that are calculated with MetaboIndicator. I will get back to this in the examples. And also these results are linked to scientific literature, so you can make additional interpretation already based on this. It's a great starting point to understand and interpret your metabolomics results to get to step 3, where you translate insights. There you develop innovative solutions to your scientific problems, from predictive biomarkers to metabotypes, risk scores, and more. So now on to our case studies where these 3 steps were applied in 3 disease contexts. Our first case study has to do with the underlying condition of most chronic diseases, inflammation. By now, it's broadly accepted that the gut microbiome has deep links to health and is a strong influencer of host inflammation. What you may not know is that these effects are heavily mediated by metabolites. So what happens is essentially the bacteria from our gut microbiome use components of our food as substrates for their own metabolism. This releases metabolites that act as signals in our gut cells, in our immune cells, and beyond. For example, the essential amino acid tryptophan is one of these bacterial substrates that has gained particular attention. Here on the drawing, you see it on top of the pathway. Tryptophan is converted to a broad range of metabolites by the gut microbiome, including indole derivatives that you see on the left of the map that influence our immune system. In fact, the balance between indole and kynurenine pathways is essential for host health. So when pro-inflammatory signals take over, inflammation ensues with several metabolites making their way into the blood and also serving as potent markers of inflammation for us. Here I want to take a moment for those of you in the audience who are familiar with our offering and our specialized quantitative assays. You'll know that biocrates offers a tryptophan assay. In the MxP Quant 1000 assay, we've included many pyridine carboxylic acids from the kynurenine pathway, and we even go beyond the coverage of our indole derivatives that is in our original assay. So there's going to be a lot of interesting new things in this assay for you if you're interested in these pathways. But so now, how can you leverage this data? How can you make sense of this information to understand what's happening in your experiment and draw interesting conclusions for your experiment? One way to do this that is very efficient is MetaboIndicator, as I mentioned previously. What this module of our software does is it automatically calculates sums and ratios of metabolites that were quantified in the sample. So when screening your samples with the MxP Quant 1000 assay, you can opt to receive these sums and ratios together with your absolute concentrations of metabolites. Not only does this provide additional data points, but it's also a great starting point for interpretation, with each indicator coming with a set of literature references to get you started on your interpretation journey. Now here, in the context of inflammation and tryptophan metabolism, a popular indicator is the ratio of kynurenine to tryptophan. We've discussed this many times in previous webinars at Biocrates. It's typically increased in the blood of individuals with inflammation. And in the white paper that I mentioned previously, we've seen the kin-tryp ratio increased in many chronic diseases, including IBD and depression, for example. But MetaboIndicator is applicable to many other parts of the pathway, and other steps of the kynurenine pathways are addressed by other indicators, which allows to start to pinpoint and understand where the pathway may be compromised or may be affected in your experiment. Once you've leveraged your data and identified microbial metabolites and inflammation signaling as relevant in your study, such findings can be translated in many ways, depending on the goal of the study. For example, for preventive medicine, these findings can be easily used to design a dietary intervention or a supplementation regimen with probiotics, that's live bacteria, or with postbiotics, that's the bacterial metabolites that are supplemented to modulate the effects of the microbiome on health, but especially on inflammation. Another context is predictive medicine. Here we have a recent example published in Nature that showed that the microbial metabolite 3-IAA, or 3-indole acetic acid, was predictive of drug response in a small cohort of pancreatic cancer patients. From there, it's easy to jump to precision medicine applications, where this same metabolite can be evaluated as a targeted chemosensitizer. That's a supplement that's given to enhance drug response. So this concludes our first case study, where we've seen that metabolomics could be applied to better understand the role of the gut microbiome as a gatekeeper of inflammation in the host, and also concrete solutions that can be developed from that knowledge in the context of 5P medicine. In our second case study, we dive deeper into energy metabolism, a common culprit in chronic disease and particularly beloved in the fields of oncology and neurology. Over the years, the core pathways of energy metabolism, such as glycolysis and TCA cycle that are heavily described in textbooks, have been deeply connected to other metabolites and pathways that feed alternative substrates into these energy-making machines, besides sugars and fatty acids. For example, amino acids, but also carboxylic acids, can be effective substrates for the TCA cycle. But what does this have to do with Alzheimer's disease? Well, in Alzheimer's disease, neurons have difficulty using glucose as an energy source. That's a phenomenon known as hypometabolism. And this leaves space for other energy sources, such as amino acids and ketone bodies, to help produce energy in those cells. In human blood and in also cerebrospinal fluid of a rat model of Alzheimer's disease, amino acid levels were reduced, suggesting a compensating mechanism through inclusion of amino acids into the TCA cycle. Of course, one may learn this in the literature, but a much easier way to figure this out is with MetaboIndicator, our tool to calculate sums and ratios of metabolites. For example, to leverage these metabolomic screening results, the ratio of TCA intermediates, azocitric acid and alpha-ketoglutarate acid, to the amino acids aspartic acid and asparagine can be indicative of a switch to amino acids as an energy source. Interestingly, in Alzheimer's disease, individuals with the ApoE4 genotype that is recognized as a risk factor for Alzheimer's also have different levels of amino acid levels compared to other individuals. So this points to the possibility that this alternative energy source could really be relevant in Alzheimer's disease. So what can we make of these findings in the broader context of 5P medicine? Here again, this all depends on your goal. So scientists have looked into predictive medicine approaches, for example, using amino acids and other alternative energy sources as early stage markers of Alzheimer's disease. For preventive medicine, supplementation with amino acids has been evaluated to help support compensatory mechanisms and avoid depletions for other pathways. And in precision medicine strategies, these have focused on designing personalized interventions with tailored dietary plans to palliate amino acid deficiencies. This concludes our second case study, where we've seen that screening samples with a broad metabolomic panel covering core energy metabolism pathways, but also alternative substrates from other chemical classes, provides a rich landscape to investigate potential switches in energy sources and solutions to remedy them. And this takes us to our third and last case study, where we look into one of my favorite metabolite classes, polyamines. Polyamines such as putrescine, spermine, and spermidine serve many functions in the body, from cell growth and nucleic acid stabilization to regulation of gene expression, and, importantly for this case study, regulation of the immune system. In particular, polyamines have been shown to be both anti-inflammatory and immunosuppressive. Polyamines are acetylated by the enzyme SSAT, which enables their transport out of the cell. And these acetylated polyamines have associated with several forms of cancer. One reason for the elevated levels of acetylated polyamines in cancer is an increased expression of the enzyme SSAT in cancers such as breast cancer, lung cancer, and prostate cancer. Interestingly, when the levels of acetylated polyamines increase, the levels of non-acetylated polyamines also tend to increase because they're being replenished by other mechanisms. So overall, this leads to an increase in the total pool of polyamines, which enhances their immunosuppressive effects. Here again, knowing this, the relevance of polyamines in cancer research seems obvious, but how could you, as a non-expert, if you are a non-expert, reach this level of understanding quickly? Using MetaboIndicator, of course. Here again, the calculation based on these metabolites can help to identify and even quantify changes in polyamine metabolism. So to leverage this data, one may use the ratio of acetylated polyamines to non-acetylated polyamines that you see represented here. Interestingly, this ratio has correlated with the levels of SSAT enzyme expression, which is encouraging for further interpretation. Also interesting is that with the type of absolute quantification provided by the MxP Quant 1000 assay, following or comparing the absolute concentrations of acetylated polyamines in blood or in urine would also be indicative of changes related to SSAT activity and other parts of this pathway. I want to take a moment also to explain the graphic that we have on this slide. You've noticed that before the drop was red because the ratios were relevant, especially in blood samples. Here, this ratio is especially applicable in the urine, which is why the color is different. And so once you've acquired this knowledge, the ideas for translation abound. With a focus on predictive medicine, the relevance of polyamines to predict immunotherapy success in patients has been tested. And consequently, in precision medicine, inhibiting the enzymes that lead to de novo polyamine synthesis is also investigated as a promising option to increase the chance of survival in cancer patients, especially the ones who have a low expression of the SSAT enzyme. In this third case study, we've seen the deep interconnections between metabolism and drug response and the added value of using metabolomics and metabolite concentrations in addition to develop new therapeutic options. I hope that these 3 case studies have given you ideas for your own research of how you can screen samples with broad metabolomic panels, leverage the results with metabolite sums and ratios to gain instant insights for effective data interpretation, and translate these insights into innovative solutions to your research problems. I expect that this first part of the webinar has gotten you excited and curious about the MxP Quant 1000 assay. So without further ado, I'm delighted to give the floor to my colleague, August Langsdorff, who will tell you everything there is to know about MxP Quant 1000 and how to implement it in your own research. Thank you for your attention.